right. Shall we start? All right. Welcome everyone to the summer 2021 ES&T Global Webinar, Bridging the Gap, Linking Analytical Chemistry to Biological Effects. I'm Greg Lefevre, an ES&T Early Career Board member from the University of Iowa, co-hosting with Susan Richardson, an ES&T Associate Editor from the University of South Carolina. Our fundamental question is this: can we link the exposure of chemicals in the environment to effects in organisms? This is one of the driving questions that founded environmental science and technology over 50 years ago, but continues to be relevant today with the proliferation of emerging contaminants in our water, air, soil, and food. The tools of today are allowing us to probe this question like never before. Advances in analytical chemistry, such as high-resolution mass spec, have advanced targeted and untargeted detection and enabled metabolomics and proteomics. Molecular techniques like genomic and RNA sequencing are allowing us to observe specific biological effects, such as developmental and hormone regulation, like never before. Thus, we stand at a convergence of two omics revolutions that are enabling us to ask questions that would have been impossible just decades ago, like novel pathway elucidation and complex mixture effects. The goal of today's webinar is to present cutting-edge research and examine new frontiers in this age-old question. We have three fabulous speakers today that we're honored to have: Dr. Tom Young from the University of California, Davis, Dr. Rebecca Clapper from the University of Milwaukee, or the University of Wisconsin, Milwaukee, and Dr. Michael Plua from the University of Illinois, Urbana-Champaign. You're muted.
Sorry for that. So my name is Susan Richardson, and I have the pleasure of introducing our first speaker for this special webinar. Thomas Young is Professor of Environmental Engineering at the University of California, Davis, and he specializes in measuring and modeling the fate and transport of contaminants and in assessing their environmental impacts. His recent research focuses on applications of high-resolution mass spectrometry in environmental monitoring and remediation to identify unknown contaminants of potential health concern. He has applied these tools to assess the quality and safety of drinking water, wastewater, ambient surface water, sewage sludge, and biomethane, and how these can be improved. Thomas has a bachelor's in chemical engineering, a master's in public policy, and a PhD in environmental engineering. He currently serves as the Associate Director of the NIEHS-funded UC Davis Superfund Research Center and is serving on the editorial advisory board for ES&T Water. He previously served on the Environmental Engineering Committee and the Hydraulic Fracturing Research Advisory Committee of the US EPA's Science Advisory Board. Welcome, Tom.
Thank you for the introduction. So echoing what Greg just said, first, I'm glad that you're here with us today and wherever and whenever you're watching this. The fundamental question that underlies the research that we're doing in my lab right now is, how do we isolate and identify the compounds that matter in environmental samples? So there's a disconnect, I believe, between what the public and policymakers want to know about environmental safety and what environmental chemists and engineers typically answer. The question that the public is asking us is the air and water that we breathe and drink and the soil that we come in contact with safe for people and the organisms that live in the environment? And this is happening in the context of all sorts of complex activities and processes in the environment. So wildfires, groundwater recharge, biosolids applications are all examples of places where the cocktail of chemicals that people make get transformed through combustion, treatment, biological processes, other reactions, and all of those things create a cocktail of chemicals that we don't necessarily know what they are. When people ask us whether the environment is safe, chemists typically tell them something different, which is that the sample that we've analyzed either contains or does not contain a particular list of targeted chemicals. And the number of chemicals that have any kind of environmental standards numbers in the hundreds, depending on where you are and in what media we're talking. The number of chemicals that are produced globally on purpose is in the tens of thousands. But the number of ways in which we can combine the elements that underlie organic chemicals is, no matter how you count it, enormous. And so as a result, we cannot rely simply on measuring the things that are in the green circle in the bottom in order to tell us whether the environment is safe or not. So how do we assess environmental safety? The basic idea is that we want to combine testing for biological effects with a complete chemical analysis of the sample in question. So in the mythical Holy Grail, we identify every chemical in the mixture, and we know what impact it has on whatever receptor organisms we might be interested in. And then we connect those two things to identify chemicals that cause the biological effects using experimental techniques like fractionation, using statistical techniques that I'll talk about today, using predictive or data mining techniques to try to identify whether the things that we've found are plausible causes of the effects that we've observed. These things are starting to show up in regulatory applications. A good example of this is the recently adopted policy on recycled water in the state of California, which suggests that requires that people monitor both for target compounds, the usual suspects in water quality, but also test the water samples to see if they mediate estrogen receptor agonism or aryl hydrocarbon receptor agonism. And there's trigger levels specified in the equivalency of some of the positive controls used in these assays. But as it stands right now, the workflow for determining what happens once you get a hit is not well established. And so our interest is in figuring out, if you have a sample that has more than 3.5 nanograms per liter of 17 beta estradiol equivalent activity, what are we going to do? And how is it going to contribute to the complete assessment of environmental safety in these samples? Obviously, I have a very short time today, so I'm going to try to do my best to summarize a lot of work that we've been doing. All of the work that we try to do either starts with an aqueous or a solid sample, either a water or drinking water or a sludge or a house dust, and does some sort of extraction or concentration, concentrates the sample, filters samples, and then goes to one of two instrumental platforms, either liquid chromatography with quadrupole time of flight mass spectrometry and or gas chromatography with the same detection capability. And I've summarized some of the publications that have come out recently and the ones that are in process with respect to different media. So our goal is to be able to take any environmental medium of interest and run it through this screening process to understand what chemicals are there and whether they're connected to biological activity. Once we've acquired the data, the workflow looks something like this. We take our data files from the instrument, in our case, an Agilent instrument, and we convert them to an ABF format, and then we use two metabolomics generated software packages that are open source, MS Dial and MS Finder. These were both developed at the West Coast Metabolomics Center at UC Davis in collaboration with the Riken Institute in Japan. And the publications that underlie them are shown here. So when we're done with this process, we have an aligned set of data across all the samples in the set that we're looking at in this particular moment. Sorry. My timing is not showing, so I can't tell where I am. The workflow that we've developed has unified our approach to handling both LC and GC high-res data. And this is the primary reason that we've selected this particular workflow, is it handles both our GC data and our LC data. And when we integrate it with the MS Finder in silico fragmentation program, we can elucidate structures using targeted MS data on the compounds that are of concern. So I want to just walk through one example that we've recently completed on this workflow that shows how it works and what we find and what the advantages are. So we did a study in the aftermath of the fires in 2017 in Northern California. We picked 5 sites. One of them, the yellow triangles up at the top of the map, are from a state park, an undeveloped area, and we took ash samples from 3 locations within that park. And then we chose 4 neighborhoods that were completely destroyed by the fire and took ash samples from 3 locations at each of those sites. So we have a total of 15 sites, and we analyzed them using the workflows that I described previously. So I'm going to focus in on 2 particular bioassays. These are cytochrome P450 1A1 activation and aryl hydrocarbon receptor agonism. We have 4 other bioassays that I'll talk about in a few minutes. What we saw across in this slide, what's shown is bioassay response as a % of the positive control compound. So for these 2 assays, the positive control is 2378 tetrachlorobenzodioxin. And so 100% activity there is 1 nanomolar of that compound. And then on the left is a negative control, which is a solvent blank. And asterisks indicate statistically significant differences from the negative control. And you'll see that some of the undeveloped sites had as much activity as the developed sites. So the homes that were destroyed are not unique in their biological activity, but we wanted to know what compounds were responsible for these agonism results. So it'll show you what an MS-dial output looks like. This is just a sample shot of the GC-EI alignment example. 595 features were identified or selected out of this annotation, and 400 of them were annotated with structural information. Some of these were statistically significantly related to at least one of the bioassays. So just to show you the summary of the alignment results, as I said, we have 3 different platforms: GC with electron ionization, LC with ESI positive and negative modes. And then we report how many of those features are statistically significantly correlated with each of the 6 bioassays. The 3 on the top, CYP1A1, IL-8, and COX-2, are all macrophage cells and measuring genetic expression. And the AHR, ER, and AR are agonism in the KLOX bioassay. And so the numbers in the table represent positively, negatively correlated features of the, say, in the GCEI, the 407 features that passed our blank and signal to noise filters. And we also report elastic net regression results. It's a regularization method that selects features from these kinds of big data sets. In the end, for GCEI, we had 76 features in GCEI that were correlated with at least one of the bioassays or selected by elastic net regression. Many of those compounds, over half, were what I would consider usual suspects. They were aromatic hydrocarbons that would be totally expected to activate the aryl hydrocarbon receptor. Some of the suspects that we found were what I would say are less usual. They are compounds that are plausible activators of AHR, but would not have been known to cause these effects a priori. So how do we know that this technique really works? And this is the place where we really evaluate success. And we believe we were successful, and I'll show you why. So in the GCEI data, we went to the TOX21 database of activity for these AHR agonists. And of the 407 things that we annotated with a structure, 15 of them were known to be active according to Tox21, less than 4% of the universe of compounds. When we zoom in on the features that were either correlated with the activity or were selected by the elastic net regularization technique, we have a much better success rate in the 20% domain. But much of the non-active stuff is just things that we weren't able to prove to be active in this using the Tox21. So we expanded our database under support from NIEHS. We've taken over 300,000 compounds and run them through the aryl hydrocarbon receptor assay. That's been done by Mike Dennison in the past, and we've now compile that into a database, and we could look through that database, and we had many more active compounds. And so now the success of these techniques, the correlation and the elastic net, are north of 70% in terms of identifying bioactive compounds. In some cases, we had to buy these standards. So the 5 compounds on the far right here were things that weren't in either the Tox21 or the UC Davis database. We bought them and we implemented them through this technique so that we could test their aryl hydrocarbon receptor activity. And now the elastic net regression was able to be 100% successful in isolating bioactive features. This doesn't say that we didn't miss things that were bioactive. We can't prove that, but it does suggest that these techniques can allow us to fish out of a complex mixture biologically active compounds with a particular biological endpoint. A fly in the ointment, so to speak, a complication is that these are agonism tests, that is binding to the receptor and activating it, not blocking the receptor from being activated by hormones that should. And so we've done some sampling with sludges and biosolids, and what we find is that there's a very strong correlation between the decrease in agonism observed across samples and the increase in antagonism. That's measured as, in the case of the estrogen assay, estradiol being present but not showing agonism that it should, because compounds in the sample are blocking that activity. And because agonism and antagonism related compounds are present in this mixture, simple assessment of causation can be confounded, and we're working on ways to untangle this at present. So just to summarize and to turn it over to the rest of the speakers, we have validated non-target workflows for a bunch of different sample types. We have shown that we can use a variety of experimental, statistical, and predictive techniques to connect unknown chemicals with their biological effects. And unfortunately, biological complexity, even for these relatively simple in vitro systems, makes it really hard to tease out cause and effect. And I think the biggest point that I want to make today is that we chose a set of bioassays that were effectively available to us here at UC Davis. And I think they're a good choice, and I think they're important bioassays. But what we really need is some sort of systematic ways to produce a set of minimum protective bioassays to assess the safety of the samples. And in my experience, chemists often don't ask good questions about what bioassays to use, but toxicologists don't necessarily want to be pinned down on which assays should be used, because there's always one other biological mechanism that could be of concern. And so I think the conversation here today is a good one to get this going and started. So just to acknowledge our funding sources, a lot of this work has been done under the auspices of the UC Davis Superfund Research Program funded by NEHS. Our wildfire work was specifically funded by an R21 award. And then the sludge work was done by my former postdoc, Gabby Black, in the NSF GRFP award. And I've got a lot of great collaborators on the biological side and on the metabolomics side. Thank you for your attention.
Excellent. Thank you so much, Tom, for that inspiring presentation. And it's my pleasure to introduce our next speaker prior to our final panel discussion and our we have two more speakers. Rebecca Clapper is professor and director of the Great Lakes Genomics Center at the School of Freshwater Sciences at the University of Wisconsin-Milwaukee. Professor Clapper studies the potential impact of emerging contaminants, such as nanoparticles and pharmaceuticals, on aquatic life and how we may design these chemicals to have the least environmental impact. In addition, she examines the transport of these chemicals through the wastewater treatment systems and into the environment, how different treatment technologies may remove them from the waste stream. Her research also links the impacts of these chemicals on the health of aquatic species to that of human health. Professor Clapper has received a Fulbright scholarship to the UK in 2017 and a AAAS Science and Technology Policy Fellowship. She served as invited scientific expert to both the US National Nanotechnologies Initiatives and the International Organization for Economic and Cooperative Development panel on nanotechnology, where she has testified on the potential impacts of nanoparticles on the environment and the utility of current testing strategies. She has also served as a technical expert to the Alliance for the Great Lakes and the International Joint Commission regarding the potential impacts of pharmaceuticals and personal care products and other emerging contaminants on the Great Lakes. She has served in the National Academy's panel on the environmental impacts of currently marketed sunscreens and potential human health impacts of changes in sunscreen usage, and another to develop a research strategy for environmental health safety aspects of engineered nanoparticles. Professor Clapper received her PhD in ecology from the Institute of Ecology at the University of Georgia. Please welcome Dr. Rebecca Clapper.
Thank you. Let me share my screen here. Okay. So, and my angle is linking some of the chemistry that you just heard to some of the biological effects that we see in organisms in the environment. And Tom brought out some really great points about how difficult this can be. You know, we are, as biologists, we're often asked to answer, are we measuring the impacts of contaminants out in the environment and natural populations? And it's a really difficult question to answer, as you can see from the milieu of chemicals that might be out there. Here's just an example. Back in 2011, the Wisconsin Department of Natural Resources had been monitoring for a couple of decades declines in perch populations in Lake Michigan. And they had attributed to invasive zebra mussels, which definitely cause an impact in the food web dynamics, and to fishing. There were all sorts of hypotheses, but one of the things that really came out was that when they collected the fish, the male fish gonads looked very strange. So up on the top right-hand side here, you see a really healthy male perch, and they're nice milky white gonads. And on the bottom, you see kind of an atrophied, weird gonad from a male perch that they had been finding basically almost exclusively back in about 2011. And then after that, we went to go collect, and they weren't finding any males at all. So it was not obviously a fishing question. It probably wasn't just a zebra mussel question. There must be some other kind of thing going on here. But the question is it any of the contaminants that we see out in the environment? And it's a very difficult question. As Tom was mentioning, you know, we have very little information on which chemicals might be causing a potential impact. Review from Wang et al. And ES&T looked at the total volume of chemicals and types of chemicals that were registered over the last several decades, and it gave a little bit of a frightening idea of how many chemicals might potentially be in the marketplace at any one time and could potentially make it into environmental systems. And so there were, and in this case, there were 3 times as many as that had been previously estimated, and many of them were publicly unknown. So we really don't have a lot of information out in the public spectrum as to what exactly these chemicals are and what they might be doing. And so trying to answer a question of what kind of impacts chemicals might cause makes this very difficult. In addition, when you're talking about organisms in the environment and you're trying to translate any results that you have in the lab to environmental reality, the reality is that organisms are exposed to single chemicals, mixtures of chemicals, and then other stressors that also cause these impacts. I mentioned invasive species, but there's also climate change. stressors that are not necessarily chemical, but are physical related, and habitat changes, as well as the chemicals that we're talking about. And so how do you separate out what impact is caused by a chemical versus some of these other stressors that are naturally being found in the environment? So there's hope with some of the new technologies, and you heard a little bit, again, from the previous speaker, about using some in vitro assays and molecular tools in order to examine what different chemicals in particular are doing to organisms, or trying to model what they do with organisms by looking at particular pathways, and then trying to link those pathways up to networks of potential effects that end up influencing the state of cells within an organism, and eventually to tissue function, organ function, and the overall adverse outcomes in organisms. And this is a long road going from these pathway analysis, or even those in vitro assays that we're talking about, all the way up to organism impacts. It's not a clear impact. If you see that, for instance, that you have an estrogenic marker causing, that's being triggered in the in vitro assays, you have to then show that it goes all the way up to an organism impact to really have this connection going between these different kinds of things. But what the molecular tools do is provide a mechanism to separate some of the impacts that you might see. So if you have several different chemicals and they're each pushing different pathways within the organism, and you have a mixture and only one of those pathways is triggered, it could be possible that you would be separating out that one chemical's impact versus others. It could maybe cause or provide some kind of answer as to what's causing the biggest impact. If there is a set of networks that are dominant in the impacts that you're seeing within a mixture, it could give an indication that that might be what's causing the greatest impact. And it also provide a way of predicting potential impacts of new chemicals that you come across that may have similar structures or functions within the organism. And so there's the hope within this ToxCast system and the toxicity testing in the 21st century that EPA and NIH have put forward and started a huge effort towards, that it could provide some predictive capabilities of what's going on in the environment. When I first started, however, we were looking at, the real world example, going back to the perch, of what could be causing the effects of on these perch in Lake Michigan, if we were just considering the chemical impacts. And what we did is we went and we measured some of the pharmaceuticals and other emerging contaminants going out into Lake Michigan, which is a large water body. But I will say that the perch tend to stay near shore for spawning, and spawn right near one of the sewage outfalls for the city of Milwaukee. And so they end up getting a higher concentration probably than of exposure than if you go out to the middle of the lake. But the thought was that this is such a huge water body that we really wouldn't find anything. And so in order to really show any kind of environmental impact, we wanted to take real world concentrations back to the lab. What we did find, which was surprising at the time, but is becoming less and less surprising, is that metformin was one of the major contributors to the contaminants that we saw going out into the lake. Each of these little boxes of yellow here is 25 nanograms per liter of metformin in particular. And so it was a bit surprising because before that time, I hadn't really ever heard of that medication. And usually people were monitoring caffeine for wastewater going out into the environment. And we did find that in good concentrations. But, the previous adage was that, these things are in such low concentrations, they wouldn't cause an impact on organisms. However, when you consider the collective discharge going out into this water body per year, it's actually fairly large and it's consistent. It's not something that goes away, and it's probably why we're measuring at higher concentrations than we had expected. However, these things are not being found at therapeutic doses for humans. They don't reach toxic levels. And so the question is, do they cause any effects? And we really don't have enough information to say completely what's going on with these chemicals when they hit organisms out in the environment. So one of the approaches, and this is still a very valid approach, is a chemical by chemical approach, looking at the chemicals that were in highest concentration and in the largest frequency of samples. And here is just an example of the experiments that we did for metformin, where we looked at the endpoints that would happen in this case, fathead minnows, when they were exposed to environmental concentrations at the highest concentration, and in subsequent experiments, some of the lower concentrations of metformin found in the environment. And some of the things we find are some adverse outcomes where we see changes in body size, changes in gonad structure, where we saw some intersex fish. And then we also see a difference in reproductive output in these organisms. And so then we take it back and say, well, what could be causing these impacts? Metformin doesn't resemble known endocrine disruptors. It's not predicted to bind to the estrogen receptor, according to those toxicity in vitro tests. However, it is prescribed for hormone-related disruptions, particularly in women for polycystic ovarian syndrome, where those women are known to have lower progesterone levels. And this helps with fertility issues and also weight and other things. And what we found is that it pushes particular parts of endocrine pathways that end up leading to changes in vitellogenin, estrogen receptor expression, and other things within the particular brain, gonad, liver pathways that are related to reproduction in fish. And so we do have evidence that this chemical may be one of the culprits, but is this single chemical a culprit? And I'll say also that since then, other folks have found similar things in other fish species. So it's probably not just this fish that's affected in this particular way. So if you look at the whole mixture of chemicals, however, it's possible that there may be multiple chemicals that would be causing this, because it's not found at the highest concentrations at all different parts. And so if you took the most prevalent chemicals, and this was an experiment by former postdoc Jordan Prego, looking at how mixtures of these chemicals would impact these organisms, and mimic the mixtures that you found at each of the locations, going out from the sewage treatment plant all the way out to the east and to the south, the dominant weather and water patterns for this particular area of Lake Michigan. And what we find is that the individual chemicals don't necessarily tell you that each of these areas would cause some kind of impact on particular gene expression associated with those endocrine pathways in fish in particular. But other vertebrates as well. But when you look at the mixture of the chemicals, there is some indication. These red numbers here are the ones that were significantly different than the control. And these are the fold changes in each of these genes, showing that there's some significant changes in these important genes along that pathway when you consider a mixture. And so, but the question arises for this, what we did is we identified certain pharmaceuticals and then picked them to make our mixtures. You could also do this by taking an environmental sample where you'd have a lot of things that you hadn't measured as well. But the question is, instead of doing a single chemical approach or doing this a selected mixture, is there a way of doing this in a better way that really looks at large numbers of chemicals and dynamic mixtures? And so here enters again the ToxCast database. There are folks using this, and we've started to as well, to evaluate if you can use some of the high throughput testing data that's available for individual chemicals in looking at mixtures that you find at contaminated sites. This is an example from the US Geological Survey in West Virginia by Rose et al., looking at all the chemicals that you find in a particular watershed, and then using the activity ratio from all of the ToxCast assays that they found that were relevant and that also had adverse outcome pathways, showing that they were related to some kind of biological event on the other end. So weren't just floating out there as just an in vitro assay. Could you predict which part of the watershed would be most impacted by those chemicals? And so this is a really interesting way to then go and narrow down where you might go sample and look for potential impacts of those chemicals, and potentially have a relative control site versus a heavily impacted site, and start to narrow down which analytes might be causing the greatest impact. So it's an interesting, relatively new way of trying to ask these questions. The drawback of the in vitro test, of course, is that it's not necessarily a clear indication of what's going to happen in an organism. And so I just want to preface this by saying that these in vitro tests are really great as possibly the first screening tool. However, they don't necessarily equate as far as the concentrations that cause an in vitro effect and an in vivo effect. In fact, the concentrations that cause or the chemicals that cause an in vitro effect might not necessarily cause an in vivo response. The molecular changes that you see could be adaptive in the organism. There aren't enough of the pathways really developed within vitro systems to really talk about what's going on as a total in the organism. And there's not a clear link for a lot of them to these apical endpoints that we care about in ecology: survival, development, and growth of an organism of interest. But it could be an excellent screening tool and really provide indications of which waterways and organisms we really need to go focus on. One of the other ways that we've been tackling this question and looking at environmental samples is using next generation sequencing, RNA-seq approaches. And the benefits of this, and here you're looking at global gene expression patterns within the organism, where you have unbiased measurements of processes going on in cells. There aren't any predetermined pathways where you have these, I'll say the in vitro assays have a lot of things associated with endocrine pathways, which are important, but there are a lot of other pathways that could be important within the organisms that cause those adverse outcomes. You can look at specific organ and tissue responses and provide a detailed analysis of multiple pathways and how they interact. Drawbacks is that when you do these samples, they're just a snapshot of time. And so really to get a full picture, you have to look at maybe changes over multiple development stages or over multiple time periods, multiple tissues to really get a full picture of what's going on. Again, these are also not tied to a final adverse outcome. You have to do more research to really make that connection. There's large quantities of really noisy data because it's a lot of data. Often there's not enough information for the organisms that we really monitor in ecological processes in the environment. And so sometimes we're limited a bit by, for instance, that a genome hasn't been sequenced and annotated fully for, an organism that we really use a lot for our environmental models or that we care about in the environment. And there can be significant differences across different organisms. So the project we've been working on most recently, and actually this is with Greg, one of our hosts for today, and Dana Kolpin at US Geological Survey and Luke Wanowitz. We've been trying to link the chemistry and looking at some of the high throughput chemistry techniques to some of these genomic results that we get for organisms. We placed fathead minnows into streams within a wastewater dominated stream. We also took water samples and took them back and did zebrafish embryo development assays. These are representatives of a paper that we just submitted, my student Emma Mead, that's in review in ESNT. What we're looking at dozens of contaminants of emerging concern and looking to see if we can correlate those with some of the changes that we see in global gene expression patterns within these embryos and whole organisms. And what we do see in the embryo assays to begin with are clear effluent impacts. So there are clear changes in gene expression going from the site above the wastewater treatment plant to right at the effluent to downstream sites. And so it does give us some indication of some of the molecular changes that are happening within the organisms. And then the key is to then tie those to some of the adverse outcomes that you might expect on a larger scale. One of the important things that we are finding is that there are some novel pathways that provide insights into these impacts of individual chemicals and mixtures that could be new biomarkers for indicators of exposure and effect that aren't necessarily in the ToxCast sets right now and aren't necessarily common things that people are monitoring. Some of them do relate back to the actions of the pharmaceuticals that we're measuring in the systems, but some of them are broader in scale. For instance, here's just an example of some of the things affecting development of cardiac muscle and cardiac muscle function, as well as adrenergic signaling that aren't common things to be measured within these data sets. Finally, I'll say that some of the new promises of these techniques are the fact that we can provide information that links some of the information going across species. We know that some species are more sensitive than others, and we have problems right now in using model organisms to really predict what happens to sensitive organisms out in nature. And so one of our goals in our lab is to look and see how organisms relate to each other in these gene expression patterns and in their adverse outcomes. And can we make some conclusions on the molecular pathways that lead to common results in organisms or sensitivities or resistance in particular organisms? This is just an example of a paper and some of the work being submitted and then also published in ESNT from my lab, looking at manufactured nanomaterials. For this one in particular is lithium cobalt oxide nanomaterials, where we saw that for one of our model organisms, this chironomid larvae, a change in the color of the organism that also was related to the adverse outcome of delayed adult emergence and the size of the organism. And what my students found is that there were lower levels of heme and heme proteins. And when we did molecular analyses, what we find are changes in the function of iron sulfur protein clusters and there are lots of them in organisms that have many different functions. And we find a lot of those functions disrupted. And with further work on here, we find that there's oxidation of those particular proteins. One of the interesting things in a paper that we just submitted to ESNT is looking going across these species. And what we find is that that effect exists not only in chironomids, but in Daphnia and in zebrafish, but to different extents, and different parts of the pathways are pushed. And these organisms have different sensitivity levels. So this data-rich analysis provides us some indication as to why these species may differ. That's separate from any uptake mechanisms or toxicodynamics that we're seeing within these organisms. So we're able to combine these lines of evidence, provide mechanisms to ask more questions as to why these things are happening than we were able to before with just acute and chronic assays within the lab. And so I'll leave this as a promise of things going forward, where we might be able to tie some of these molecular impacts to chemicals, and then also on the other side to adverse outcomes going into the future. And I'll just thank my funding sources. I forgot one for this project that we have with Greg. NIWR funded that wastewater dominated stream, but I've had funding from our local wastewater treatment plant, the Argosy Foundation, National Science Foundation, EPA, and others. And of course, this work involves my whole lab, not just me. And I've had lots of fantastic students and postdocs that have accomplished this work.
Thank you so much, Rebecca. That was really great. Appreciate it. So we are going to move on to our final speaker for this webinar, Michael Pleva. Michael is Emeritus Professor of Genetics and University Scholar in the College of Agricultural, Consumer, and Environmental Sciences at the University of Illinois. He was a member of the NSF Center Grant and is with the Safe Global Water Institute in the College of Engineering at Illinois. He developed a robust quantitative database on the toxicity of disinfection byproducts and currently is focused on identifying disinfection byproduct forcing agents of toxicity in disinfected waters. Michael has more than 270 research publications. He served as director of the Institute for Environmental Studies, president of the Environmental Mutagenesis and Genomics Society, and the Director of Undergraduate Research for the Howard Hughes Program at the University of Illinois. He was awarded visiting professorships at the Oak Ridge National Laboratory, NIEHS, Kyoto University, the Czech Academy of Sciences, and Bradford University. Michael was a Fulbright Senior Scholar and a Phi Kappa Phi National Scholar. He served as chair of the Gordon Research Conference on Drinking Water Disinfection Byproducts. And Michael has received many awards, including 2 Scientific and Technological Achievement Awards from the US EPA and the Senior Faculty Research Award from his college. Michael holds a bachelor's degree in biology from Loyola University, Chicago, and a master's and PhD degree in biological sciences and genetics from Illinois State University. Welcome, Michael.
Thank you, Susan. Oops. Good. So thank you very much for that introduction. And I'm very pleased to present this information from, probably a couple decades of work. I am going to primarily focus on disinfection byproducts in drinking water as an example of integrating analytical chemistry and analytical biology. One of the things I want to point out is that there is a profound difference between descriptive qualitative biology and quantitative biology. And one of the things which has been alluded before is that in biological assays, you're going to have a great deal more of error terms because the detectors that we are using are living cells, and they grow, and the suspension of cells really have a set of cells that are in different growth and metabolic states. So when you begin working with a chemist and you want to do some analytical biology, decide on your cell type and your biological endpoints and the statistics that you're going to use. What are you trying to measure? What is the resolving power of the assay? Define your appropriate statistics and understand the limits of the biological assay. Remember, a cell is not a tissue, a cell is not an organ, and a cell is not an organism. So so it is not wise, I believe, to try to predict from a cell to human health effects, although cellular assays are very useful in defining the range of toxicity, screening agents to determine which are more toxic and which are least toxic, and well as mechanism studies. So one of the things that I find in the literature that it's a little bit vexing is when people use static concentrations instead of defining specific concentration ranges for each agent that they're working with. Instead of saying, I'm going to use 0.11, 10, 100, 1,000, whatever concentration is. For each sample that you're looking at, do preliminary experiments so you know exactly the concentration range of effect that is effective, and then focus into that concentration. Use molar concentrations for individual compounds, concentration factors for complex mixtures. You're going to need a lot of replicates per sample, and use statistical power as a measure to determine the quality of your data set and when to stop conducting repeat experiments. Normalize your data to concurrent controls. Avoid using reference compounds. And this is important because if you use a reference compounds that define toxicity, you may end up not being able to compare your data with other studies that are in the literature. And also use well-defined measurements to define a toxic response, such as an LC50, an EC50, or some other type of in vitro assay or an endpoint that people are familiar with. Now, when I suggest knowing your assay, I mean that you know exactly every step along the way. The Water Research Foundation actually provided the original funds in around 2000 for our lab, Dr. Elizabeth Wagner and myself, to establish a number of mammalian cell assays to measure specific and to rank the cytotoxicity, genotoxicity, endocrine disruption, and also thioreactivities of sets of disinfection byproducts. And because we wanted to know, is there a range in toxicity that you can find with these agents that are generated when you disinfect water? And in fact, you can see a large range. And I think that this is a very useful thing because then you can start asking questions. Our database right now, we've published about 103. It's currently at about 130 DBPs, which is only a minor fraction of the DBPs that are available. But to show the precision and sensitivity of at least one of these assays, this is chronic cytotoxicity. This is a study with Susan Richardson's laboratory in which they were looking at different isomers of iodophonol. And you can see just by moving the iodine moiety, you can elicit a different range of responses. And the 2-iodophonol is the least toxic, whereas the 3 and 4 are more toxic. And so these two agents, 3-iodophonol and 4-iodophonol, are statistically more cytotoxic than the 2-iodophonol. And because the precision of our assays, we were able to show that 4-iodophonol was actually significantly more toxic than the 3-iodophonol. But this has very useful information when you're trying to establish structure-activity relationship studies and modeling. Now you can do the same thing with complex mixtures. The last slide was with a single compound. This is with a complex mixture. This is a solid waste effluent that we've been doing work with the Department of Defense. And if you take your solid waste effluent, it is rather toxic. And you can see this in these black squares. However, if you use direct particle reuse based on granulated activated carbon, ultrafiltration, reverse osmosis, and UV H2O2, you can convert this very toxic wastewater effluent into a much less toxic effluent. However, if you take that effluent and you chlorinate it, it becomes as toxic or perhaps even a little bit more than the original effluent. And so this allows you to evaluate each step along the way when you're trying to engineer a remediation or processes to remove the toxicity, in this case, of wastewaters. And it's not great to assume, it is dangerous to assume that adding an additional component on a reuse process is going to give you safer water. In this case, just the opposite happened. And so this is very useful to have these types of assays in combination with the analytical chemistry to drive you along. Now, I show this a study here. This is part of a toxicogenomics study, but I want to point out we use different types of cells to get different types of information. But I want to point out this is CHO and this is human, non-transformed human cells. I just want to point out this pattern. This is iodoacetic acid, bromoacetic acid, chloroacetic acid. So we're looking at the different halogen moiety in these monohaloacetic acids. These are well-defined haloacetic acid disinfection byproducts. But we wanted to know what the mechanisms of the induction of these haloacetic acids. So one of my graduate students, Justin Pels, did enzyme inhibition analysis on glyceraldehyde 3-phosphate dehydrogenase. This enzyme is a major enzyme in glycolysis and in energy metabolism in cells. You block GAPDH, you stop the generation of pyruvate, which then causes starvation of oxidative phosphorylation in mitochondria. You have a drop in ATP, lose cellular homeostasis, and you can get an increase in reactive oxygen species. So when we look at the enzyme inhibition studies, you will notice that we got iodobromochloride. The same pattern came out. Azradad in my laboratory then actually measured the effect on ATP in cells, and per picomole of each of the HIAs, you could see that IAA induced the most reduction in ATP, or the greatest amount of reduction in ATP, followed by bromo, followed by chloroacetic acid. So there is this link between the halogen and the response. Doing toxicogenomic analysis, we did PCR-related gene array studies, and we found that COX-2, which is in the pathway of oxidative stress, was highly affected, and this modulates the activation of NRF2. So in this case, we went from CHO now to non-transformed human cells. Once we identified that we were getting a pathway associated with real oxidative stress, we then used a recorder gene. This is a different cell line. These are HepG2 cells that are looking for the activation of antioxidant response element, and this is the major switch that cells use to address oxidative stress. And you'll notice even here we show iodo, bromo, and chloro. That same pattern that we showed in the original in vitro cellular studies is now reflected in the activation of ARE. So since we have this large database, a number of laboratories in the last couple of years have been looking at quantitative structure-activity relationship modeling using these CHO cells. And Lin Young Mo's laboratory looked at 74 DBPs. Min Teng's lab published in 2020, 15, and then Zhao Wei, also working with Min Teng in our laboratory, published 13 DBPs in which we had sufficient information to actually do high quality quantitative structure activity relationship research. Now, what this comes out to be is rather useful. So a few years ago, Susan's laboratory published a paper in ES&T in which she was looking at the development of new disinfection byproducts after chlorination of X-ray contrast agent contaminants in water. And she in her laboratory identified some new iodinated disinfection byproducts. One of them was chloroiodoacetyl Now, this is not in our database, and it's difficult to get a hold of this material. However, because of our QSAR modeling, we were able to come up with a prediction of its LC50 in CHO and also its genomic DNA damage, 50% tail DNA damage. So these models can be very useful because they can then be used to calculate out the toxicity that you would get from untested compounds that happen to be disinfection byproducts. I mentioned that we started developing these assays back in 2000 and with grants from the Water Research Foundation and from EPA. And early on, we made some interesting observations, and these graphs here are kind of descriptive. They're just showing I'm jamming together cytotoxicity and genotoxicity index values. But the take-home message here is that the carbonaceous DBPs are much less toxic much less cytotoxic and genotoxic than nitrogen-containing DBPs. And then iodinated DBPs are more cyto and genotoxic than brominated DBPs, than chlorinated DBPs. And this kind of information is very useful, especially since we do not regulate any nitrogen-containing DBPs, nor do we regulate any iodinated DBPs. So this is what I look at as being a very nice collaboration in terms of analytical chemistry and analytical biology. Susan Richardson's lab and my lab analyzed 13 city waters from a large study funded by the European Union. And Susan's laboratory would take a water sample, concentrate it, and then do chemical analysis of 100 or so DBPs, and she would get this distribution. Our laboratory was doing the analytical biology, just as happens to be cytotoxicity, but we also did a lot of genotoxicity work. And by using just chemical distribution, you would look at this and say, okay, the THMs, the regulated DBPs, the THMs and haloacetic acids account for 45% of the toxicity because that's based on the distribution. However, if you take these data and then integrate it with our toxic potency values, and we call this approach TikTox, it's a chemical, it is a calculated measurement of not only concentration, but of the toxicity of the individual compounds. And if you now look at that distribution, we find that the THMs and HAAs only really contribute about 1.5% of this toxicity, whereas the nitrogenous DBPs are predicted to account for about 82% of the toxicity. Now, this is kind of fun when you do this calculated wise, but it's always better to do real experiments. And so in a study, this is a series of studies that my colleague Bill Mitch at Stanford and I have been working on. And this is where we're actually looking at 8 different drinking and potable reuse waters. And we took we generated defined mixtures based on 43 regulated and unregulated disinfection byproducts. And then we did the toxicological analysis. And so this is a very large database, but I'm just taking one water over here. If you take that mixture and break it up into regulated, unregulated, and then the mixture between the regulated and unregulated, you see that we find that the toxicity induced by the regulated DBPs, and these are all summed molar concentrations, so we're comparing molecules with molecules, the regulated DBPs are relatively non-toxic. However, the unregulated DBPs are highly toxic. And when you mix them, and when you look at the complete water mixture, you get something in between. When you stand back and from this study, we also identified that DBP mixtures essentially for cytotoxicity was additive. When you stand back and look at the entire set of 8 different waters with their 43 different disinfection byproducts, and look at bioassay-based versus calculated toxicity-based, you find that the regulated DBPs both agree in that both approaches agree that the regulated DBPs account for a rather low amount of toxicity, whereas the unregulated DBPs account for the vast majority of the toxicity of these water samples. And so these data clearly show that the regulated DBPs are not the forcing agents for toxicity. And we are now continuing this work with genomic DNA damage analysis. And our initial data are showing that, in fact, that we're getting similar types of studies, similar types of effects, where the unregulated DBPs are much more genotoxic than the regulated DBPs, and the mixtures that combine both regulated and unregulated disinfection byproducts are in between the two extremes of the regulated and unregulated DBPs. So we're showing an effect for not only cytotoxicity, but now also for genotoxicity. So what do you do with this? Well, recently Susan and I wrote an opinion paper in which we asked that question. What do we do with the more toxic disinfection byproducts? From the science, the take-home message is that the regulated DBPs are not the forcing agents for toxicity in disinfected water. And it is the nitrogenous DBPs and the ionated DBPs that generate the majority of DBP-mediated toxicity in disinfected waters. And I think this is an important thing for regulators to understand. Clearly, in vitro assays cannot be directly linked to health. But I want to point out that some of the NDBPs that we are identifying also show up as positive carcinogens in the NTP database using whole animals. And so I think that there has to be a fresh approach when we start thinking about what disinfection byproducts does the US EPA regulate in order to protect the public health and the environment. And the take-home message in terms of my own personal thoughts are, know your bioassays. So you actually know what you're measuring and why you're measuring them, and how this relates to the analytical chemistry that is being conducted. And this is so interdisciplinary teams, as we've seen in our previous two speakers, interdisciplinary teams of scientists can work together to prevent unintended adverse human health consequences and the implementation, in this case, of new methods to desalinate, decontaminate, reuse, and disinfect water. I don't have a thank you slide with my grants agencies, but I do want to especially thank the currently two NSF grants that currently are funding us, a past NIEHS grant and a current Department of Defense grant that has been supporting this work over these years. So thank you very much.
Thank you to all, excuse me. Thank you so much to all of the panelists for everyone today. This is a really wonderful discussion and a set of presentations. And we're going to take time for a few questions and things here. So I think we'll start off first with our last speaker. This is a question directed towards Dr. Plua. How do you validate your Q-stars with the DBPs, and what type of error do you typically see between your experimental and the theoretical results? Oh, you're muted.
So the way that we have approached that question is, yes, the Q-stars are based on the databases that we've generated over time. But then when you do define mixtures, you're actually asking that question directly. That's why when we approach it, the question of forcing agents, we do it based on calculated toxicity and structure activity relationship work. But then we take those individual compounds, make the mixtures, and then do the bioassays. And we ask the question, are they close? And in most cases, they're very close. The widest ranges that we're finding is maybe a 12% difference, which when we're considering we're working with large numbers of DBPs, that's a very small difference. So the alignment of the data actually surprises me. And it depends on how careful you are in terms of working. The greatest source of error is pipetting. So that's the truth.
Okay, great. Well, I'll go ahead and ask the next question, and Tom, this one's for you. So Tom, beyond antagonism, are you seeing any other matrix effects that affect bioassay response? And then if so, what are they, and what are you doing to control these matrix effects?
Short answer is that it's very hard to tell when you see these effects, right? Because we see, we have multiple samples in a sample set, and we measure the biological response in these in vitro assays directly. And because there's this complexity going on. There's no good way to know that the background matrix has or hasn't influenced the response in an important way. And I think, what's important with all this stuff is to be aware of what you can't do and what is limiting it. I feel like what we've been spending our time trying to do is to just convince ourselves that this approach can successfully find and identify things that are of importance. What we can't do at this point is say that we have found the most important things in the mixture, or that we haven't missed any things in the mixture that are important. And I think that, that's where we're at. But I think it's important to contextualize this as, I view this as being a way to improve safety. I feel like Michael's talk was perfect in this way. We have this list of candidates that we know we're worried about, and we regulated them. And they aren't unimportant, but they may not be most important. And if we start doing something in this domain, we can expand our coverage of compounds that we're paying attention to, and we can do better over time. And I think there's this risk of letting perfect be the enemy of the good. If we wait for the perfect set of bioassays, we're never going to have them. And so if we choose some wisely and we add that onto what we're doing right now, and we prove that we find some things that are of concern, then we will have done environmental good, and over time we can refine that. So that's what I hope. And I feel like we get into these places where we're like, we can't do anything because we can't do something that's perfect. That's my soapbox.
Thank you. This question is directed towards Rebecca. Is how do you account for bioavailability in your analytical sample measurements and linking that to corresponding bioactivity data?
Yeah, that's a really good question. Yeah, there have been a couple of studies showing that some of the gene expression profiling really relates to, when you're looking at different compounds, it really relates to how those compounds are taken up by the organism and into particular tissues where they have activity. And so one of the things we've been doing with Greg actually is trying to analyze the fish that were in those streams and looking at which of the pharmaceuticals, the 50 pharmaceuticals that were most common, do we find in the fish tissues? But it's it's not an easy task, as Greg will tell you, to do within tissues, especially they're even lower concentrations sometimes, sometimes higher than the environmental samples that they're found in. So it's not an easy task. In that example I gave, where I was looking across the different organisms, looking at the lithium cobalt oxide nanomaterials, we did look at uptake. And what we found, which kind of surprised us, was that the gene expression patterns that we saw were not related to uptake. It wasn't that the organism that picked up the most of the nanoparticles, the Daphnia, had the greatest gene expression changes that happened within the organism. It was actually the zebrafish. And so it's not necessarily related to uptake in that case. It really has to do something fundamentally different about the physiology of those different organisms, and which makes it even more interesting, right? It's not just an uptake question. It's how do those organisms differ in their physiology and in their their metabolism and their gene expression in order to cause those changes we see in sensitivity. So it can lead to lots of interesting questions. A really good point, though.
That's great. Okay, I'll ask the next question. So Michael, you're up next. And I think this is not what Greg asked you before, but what methods do you use to validate the QSARs for DBPs? And what percentage error range do you typically find for the QSAR results versus your experimental results?
I don't quite understand the question, but what I will say, the QSARs provide us a way of predicting toxicity when you use a large database to identify for predictions. So in other words, the validation would come from the standpoint that you're able to have the empirical biological data, use that in a QSAR to predict what another types of untested samples would be. And in that case, we have essentially done that. We've actually looked at our QSAR projections and have actually, they're not my QSAR projections, they're from the literature, and we've gone and analyzed new DBPs that we haven't had in our database, and they're relatively close. I'm rather surprised. I don't know what the percent difference would be off the top of my head. But the other thing that's really important is when you think about the mixtures, because mixtures is the big problem, I believe, when you're talking about environmental biology, when you're talking about any type of exposure to an environmental agent or a complex mixture, such as water. And in this case here, since we can go and make our own mixtures, and then we can then ask the question, does our calculated toxicity predict what we're going to see in the bioassay and vice versa? Once you get close correspondence with that, then I think you have good validation. Do you have perfect validation? Of course not, but you have good validation. Actually, more than I would expect just by being a cynical old man.
I think maybe one closing question that appears to be directed towards the entire panel is, someone is curious about the toxicant identification evaluation or effect-driven analysis or effect-directed analysis as different methods. And these, they seem to be potentially used more in international settings or something. And that could potentially be contextualized as far as like other, future directions and things like that. If anybody wants to kind of comment about any of those two specific things or places where you see as new opportunities going forward at this kind of convergent research.
Yeah, I have a couple of quick thoughts on that. What I've described is completely compatible with effects-directed analysis. You can do what we did on whole samples on fractions, and it will work probably better than working on the whole sample because it will untangle some of the effects that I was describing. With respect to TIE, it is very useful to know, and we're doing a number of studies right now where someone has removed toxicity with C18, and then you know that based on the pre-C18 and post-C18, the chemicals that disappeared must be somehow related to the toxicity. And that kind of work, again, it can be, everything that I described can be used with that even better, because now you've got a narrower list of suspects. Anytime you can narrow the list of suspects, you're going to do a better job.
Does anyone else have or want to add to that particular one? Or?
The other approach can be when you're working with colleagues in engineering, for instance, and they're trying to develop new methods to detoxify wastewaters or sludges or metalworking fluids, things along those lines, and they will produce a reasonable set of sentinel compounds in which they're going to look to see if they disappear over the processing of their samples. We've done a lot of work on carbon sequestration methodologies, and we could show that specific types of agents that individuals were interested in would disappear by a processing method. But that doesn't necessarily mean that the toxicity disappears. And that means that you can use your bioassays to help direct those types of engineering systems. Even if you don't know what the chemicals are, you can identify which of the processes are going to be more effective in removing the toxic components in the system. And I think that's a useful approach. It doesn't give you the identification of a specific compound, but it can tell you whether or not your engineering, whether your process is eliminating a toxic response rather than enhancing it.
And I'll just say from the biological standpoint, using a thing like that measures estrogenicity is very useful. We know that there are reproductive consequences for estrogenic compounds. And so having particular pathways that we already have some ties to adverse outcomes are really important. And so it's not to poo-poo those at all. I think that what we see here, though, and the promise of a lot of the things that we're talking about is the chance to expand the realm of other things that we could test. And it's not to put more burden on regulatory agencies, et cetera, but it's really to really try to better answer the question of the potential for the chemicals to impact the larger scheme of the environment. And our methods that we're using in our regulatory systems really accurate in representing what happens in an environmental situation. So it's really exciting, all of the data and all of the different techniques that are coming online right now because of the resources that we have just over the last decade. It's fantastic. And so I think it'll just end up allowing us to ask some really more interesting questions. And it doesn't have to stymie everything. It's just, it's scientists asking really good environmental questions, I think.
That's a really, really great point, and for us to end on here today in terms of thinking about the next questions that we're able to answer and ask as a result of these new tools. So on behalf of everyone in the audience, I want to thank all the panelists and the speakers today. And on behalf of all of the speakers and the hosts, I want to thank all of the members of the audience. So we hope to see you back again at another ES&T Global webinar. Thank you so much.
Yeah, thank you. And any questions that didn't get answered today, feel free to email our speakers. They've graciously offered to answer remaining questions by email. So thank you again to everyone for joining us today.